Hi friends, so now I am back with my next video, and in this video we are going to talk about the biosynthesis of fatty acids. So please make sure to like, share and subscribe my channel. So what are fatty acids and from where it is produced and why they are important? So first we will talk about what are fatty acids. So fatty acids, it's a carboxylic acid with an aliphatic chain. So here you can see different examples of fatty acids. So it contains a carboxylic group, the COOH, with an aliphatic chain, which is a long chain carbon, which can be either saturated or unsaturated. So what does that mean? Is saturated is when all the bonds of the carbon are used. So here you can see all the four bonds are used and everything is single bonded. But in unsaturated fatty acids, there are these double bonds which are present instead of the single bonds. And these double bonds make the fatty acid unsaturated because it introduces the kink like the bend structure and makes the fatty and these fatty acids are called unsaturated fatty acids. So why where fatty acids are used and what is the function of the fatty acids? So why fatty acids are used and what does this mean? So fatty acid synthesis is the creation of new fatty acids from acetyl coenzyme A and NADPH molecule. So fatty acid synthesis, it's an anabolic reaction, which means the formation of something like the formation of fatty acids, and the source is coenzyme A and NADPH. And where it is produced, it mainly produced in the steps are you that are uh, the steps that are required to produce free new fatty acids occurs in the cytosol. So the question is where these fatty acids are used. So the fatty acids, if you know, they can be like the free fatty acids, they are generated by the carbohydrates which you can get from your diet and like the glucose. So these carbohydrates and glucose, they undergo glycolysis which releases the pyruvate which can be converted into acetyl coenzyme A. So whenever there is the free glucose available and free more of the glycolysis happening, the free fatty acids are produced which can provide you energy, and these fatty acids are important like for the beta oxidation, which is like these acetyl coenzyme A, as you can see, the free fatty acids they can come back. Like if there are more free fatty acids, they can be used to produce the acetyl coenzyme A again and ketone bodies, which can provide you energy, right? Because these ketone bodies they are important for different cells like the immune cells to function and also for other cells like during the starvation, like when you don't have enough energy, the ketone bodies are the source which provides the energy to the brain. So these are also used like different ketone bodies, and then they can also undergo this citrate cycle again. So all the time there is this feedback mechanism which is going on from the production of glucose when you have excess of glucose to form fatty acids which can be stored in the liver, like which can be produced in like synthesized mainly in the liver and adipose tissue. And also when you have too much of lipolysis going on, like when your body is burning fat, then also the free fatty acids are released. So here we are not talking about the breakdown of the like during the lipolysis when there is breakdown of the triacylglycerol, the fat. But here we are talking about the synthesis of new fatty acids, which is the de novo synthesis. So the first step, which I told you that it happens in the cytosol, but the acetyl coenzyme A, it's mainly produced by the glycolysis and which undergo like the pyruvate and then acetyl coenzyme A. It occurs in the mitochondria. And from mitochondria, we need to transfer this acetyl coenzyme A to the cytosol. But the mitochondrial membrane that you can see here, it is impermeable, which means it doesn't have transporter, and this acetyl coenzyme A cannot cross this membrane. So what we need to do is this acetyl coenzyme A first needs to be converted to the citrate, the first step of the TCA cycle, citric acid cycle. And this citrate can go outside, which can again be converted to acetyl coenzyme A and release oxaloacetate. And then this acetyl coenzyme A, by the action of different enzyme, can be converted to fatty acids. So now, today we are going to talk about how the acetyl coenzyme A can convert it to free fatty acid, like that converted to the fatty acids, the new fatty acids. But this fatty acid, it can also comes from different source, as I have told you, right? From the breakdown of the triacyl glycerols and also from the building up of different fatty acids and from different sources of the TCA cycle. Also, the fatty acids can be formed. So here we are going to talk about the de novo, which means it starts from the scratch, like acetyl coenzyme. But obviously there are different sources of fatty acids as well. So first step, as we have talked about the transfer of the acetyl coenzyme A from the mitochondria to the cytosol to be available to the enzymes. So next step that we need to discuss is the formation of malonyl-CoA enzyme A. So this is the first step of the reaction. So now we have the acetyl coenzyme A, which needs to be converted to the malonyl coenzyme A. So what is a malonyl coenzyme A? It's the same as the acetyl coenzyme A, but with an extra carboxyl group. So what we need to do is to transfer a carboxyl group which occurs with the help of the enzyme acetyl coenzyme carboxylase, which transfer the CO2, the one carboxyl group COO negative, to the acetyl coenzyme A using ATP and releases ADP and PI and with the help of biotin. So with the help of biotin, the carboxyl group get transferred to the acetyl coenzyme A and forms malonyl coenzyme A. So this step is very important, and this enzyme is the rate limiting enzyme of this reaction. So if we don't have enough of this enzyme, we cannot produce more fatty acids, new fatty acids. So this enzyme is the rate limiting enzyme and is very important step to convert the acetyl coenzyme A to malonyl coenzyme A. Because in the further reaction, we use acetyl coenzyme A, but mostly what is used is malonyl coenzyme A. That I will talk later how we use this. So this step is called carboxylation. So now we have the malonyl coenzyme A and the acetyl coenzyme A. So this is called the condensation, where we are combining both acetyl coenzyme A and also the malonyl coenzyme A. But before that, so the first step that I told you that occurs by this enzyme, acetyl CoA carboxylase, but after that all the steps happen by this fatty acid synthase complex. So there is a complex of 7 enzymes which are required for this reaction. So all the enzymes are called, like in total they are called the fatty acid synthase complex, but which include different enzymes which perform different functions. So first step is the condensation of malonyl coenzyme A and the acetyl coenzyme A. So here you can see we have an acetyl coenzyme A, and then we have this fast complex, or this is the enzyme, like the coenzyme A with the cysteine group, and which is attached to the acyl carrier protein. And with the help of this acetyl coenzyme A, ACP transcyclase, you see this coenzyme A and here. So all the group of this get transferred to here, right? So this hydrogen is released, and this, like the from here the and coenzyme A, like you can see coenzyme A from here and SH from here, they get released, and this group get attached to this cysteine. So this is what we call transfer of acetyl group to the cysteine here. You can see, right? To this complex we attach the acetyl group, and it forms acetyl SACP, acetyl acyl carrier protein. So this is an acyl carrier protein to which we attach the acetyl coenzyme A. Same we do with the malonyl coenzyme A. So we have the acetyl S enzyme which is formed in the last reaction, right? It's just that in this case we have transferred this acetyl group to the cysteine. So before the acetyl group was on the ACP protein, but we need to free this because as you can see here, when we are adding acetyl, it get only transferred to the ACP group. So to add further groups, further carbon groups, we need to free this acyl carrier protein. So to free this acetyl group shifts to the cysteine. So when it shifts to the cysteine, ACP group is free, and now the malonyl coenzyme A, which was formed in the first reaction, it get transferred here with the action of malonyl coenzyme A, ACP transcyclase, and then again coenzyme A from here and SH group from here get released. So what we get is acyl malonyl coenzyme. So containing the acetyl group and containing the malonyl group. So this again, what we need is the transfer of this group, because if we start doing this, the individual reactions, it's not possible. So what we need to do is to transfer this group, the acetyl group, down to the ACP protein so that it can go further reaction. So now what we get is, so you can see here the this group get like this acetyl group get transferred to the malonyl, and the COO which we first added it using the biotin get released. So every time this CO, whenever we are doing further reaction, it is never used. It always get released. So as you can see here, the CO get released, and what we get is ketoacyl ACP, right? Okay. So the next step is the reduction. So you can see here, so this until now what we have done is condensation, which means like combining two molecules like the acetyl coenzyme A and the malonyl coenzyme, and what we get is beta ketoacyl because it contains the keto group at the beta, like here you can see it contains the keto group at the beta carbon, this one, and then it's the ACP protein, so we call beta keto acyl group here, ACP. Okay, so what now we need to do is the reduction reaction. So as you know, the name suggests reduction means the addition of hydrogen to this group. So what we now generate is with this reductase, the addition of hydrogen to this group. So now what we have is we add the OH group and it becomes hydroxyl. So now what we have is beta hydroxyl acyl ACP. So what we have, by adding the OH, we are doing the reduction, and the compound that is formed is beta hydroxyl acyl ACP because it has the hydroxyl group, acyl group attached with the ACP. So these are the two important steps of this reaction, the condensation and reduction. And the next step is dehydration. So what does dehydration means is the release of the water molecule. So now what we are doing is releasing this water molecule, OH from here and one H from here, and it becomes trans-enoyl ACP. Right, we remove the water with the action of dehydratase, and then we get is enoyl group. And if we do the reduction again, like there is another reduction, which is the addition of hydrogen, because the bond here is unstable, like it's a double bond, and we need to reduce this because the single bond is much better than the double bond. So when we add the hydrogen here, the reduction, it forms butyryl ACP. So now you can see we get a carbon, like we get a molecule with four carbon molecule, and in the same way we can transfer this because now we are done with this reaction and now we need to add more carbon molecule, and as we done before we need to free this ACP to add more carbon molecules. So what we can do is transfer this group to the cysteine and the ACP will be free. And when this ACP will be free, we can add more carbon group like the malonyl again and again to this reaction, and the same way we can form butyryl again and again. Repeat this reaction more times to the whatever number of carbons you need. So this step is called the elongation step. And once you do this, like if you have a length of like 12 to 13 or like 16 like you need, so in this case we are talking about palmitate, which is a 16 carbon molecule. And when you reach 16 carbon molecule, you need to separate this from this ACP carrier protein, right? The ACP group. So what we do is called palmityl thioesterase, which broke the ester bond, which means here thioester bond with the thiol group, right? SH is released, and then we get a free palmitate. So if you need more carbon molecule, you need to repeat this step more and more, because if by adding this, we are adding every time two group, because we can add here malonyl again and then again malonyl. So in every carbon cycle, in every like this cycle, you are adding two carbon. So if you need like we have four, and if you need like 12 carbon molecule, then you need to do this reaction four more times to add eight more carbon groups. In the same way, you need to do this as much as carbon you want. So in the end, we can separate this using the thioesterase and we get a free palmitate. So for this 16 molecule, what we used? We used 8 acetyl coenzyme A, right? 7 ATP, which required for the conversion of acetyl to malonyl. Why 7? Because we need in the beginning 1 acetyl, right, for the condensation, and 7 malonyl. So this in total 8 acetyl, but 7 of them get converted to malonyl. So we need 7 ATP. And then what we are using is NADPH, because there are two reduction steps, and so it's 7 cycle, because in the first cycle there is already 4 carbon. So 14 NADPH which are used and they are released. And then 8 coenzyme A are also released in the first step, because every time you do this, you release one coenzyme A, because it get transferred to the ACP group, and during the condensation it releases coenzyme. So in the overall, you need one acetyl coenzyme A, 7 malonyl, NADPH. So from where does this NADPH comes is the hexose monophosphate pathway, if you know. So it's the conversion of glucose to the shunt cycle, which releases more NADPH instead of NADH. So for fatty acid synthesis, we require mostly NADPH. And if we don't have enough NADPH, this cycle cannot happen like as efficient as we need. So in the overall, we know what this reaction is, how it works, and what are the mechanism. So what are the feedback system that can affect this system? So first, as we have talked about this enzyme, acetyl coenzyme carboxylase, which is very important for the conversion of acetyl to malonyl coenzyme A, because without malonyl coenzyme A, we cannot proceed with this reaction. So this is the rate limiting step, and if we don't have enough of coenzyme A carboxylase, this is not possible. So it will decrease the fatty acid synthesis. The second is citrate. So if we have lot of citrate present in the system, the citrate get converted to the acetyl coenzyme A, as I have talked before, which can promote the fatty acid synthesis. So this is a positive feedback system. And the another one is palmitoyl and malonyl coenzyme A. So if we have tons of malonyl coenzyme A and palmitoyl coenzyme A, the system senses it, and then there is a negative feedback system to decrease the acetyl coenzyme A, and thus decrease the fatty acids. So I hope this is clear, and I hope you like my video. And if you have any doubt, please let me know in the comment section, or if there are specific topic that you want to know, also please let me know in the comment section. Please like, share and subscribe my channel. Thank you.